Forensic intelligence has been making extensive use of human DNA since the late 1980s to link DNA evidence with its owners. Environmental DNA, eDNA, the DNA of everything, emerged decades later. While it has transformed ecology, it has yet to be applied in forensic casework, despite its vast potential to establish stronger and broader associations. eDNA refers to genetic material that organisms shed into their surroundings through skin cells, feces, saliva, mucus, hair, or decaying tissue. eDNA is ubiquitous because organisms, especially microorganisms, are present everywhere. However, it is not homogeneous or uniform. It is unique to people, places, and objects. By collecting eDNA samples from the city, people, and objects and combining it with existing eDNA data, we built a library of the eDNA of everything. Only through interactions is biological similarity created, which lasts from several days to several weeks, long enough to collect the evidence. In the following, I will explain how eDNA can be collected from the crime scene, sequenced, analyzed using AI tools, and processed to aid in solving crimes. At the crime scene, disturbed areas likely hold eDNA evidence. This evidence links the offender, crucial for our investigation. I'll swab these sites carefully, ensuring no evidence is missed. This scene has a bullet shell, significant evidence. I will swab the shell to collect the eDNA evidence that is necessary to connect the offender to this. The sealed sample is ready for lab analysis to solve the case. The eDNA collected from the crime scene is sent to a DNA sequencing lab. There, the eDNA will be extracted and sequenced using next-generation shotgun sequencing. This approach will capture the DNA of every single organism from the crime scene. Trees, plants, birds, animals, humans, and of course, microorganisms. They would all be captured. In our lab, we will analyze the eDNA sequences returned from the sequencing platform. Each sequence is compared against a vast database containing the DNA of all known organisms. This way, we can identify all the organisms in the original sample and calculate their abundance. Now we can predict things like shooting time from the bacterial growth patterns, objectively and accurately. By comparing the organisms in the eDNA sample to the eDNA data in our library and their abundance, we can offer a wide range of solutions that connect people, objects, and places. Masking eDNA evidence is a lot harder than manipulating DNA or fingerprints. In fact, it is impossible. Finally, matching the eDNA from objects found at the crime scene to the eDNA of suspects can be done. Such a comparison will be as accurate as using STR techniques. By combining vast biological databases, powerful AI algorithms, and next-generation sequencing, we can now explore for the first time the huge potential of eDNA for forensic intelligence.